Thank you so much, everyone, for coming here. And it's my pleasure to introduce Professor Kibria, who is from Morehead State University, Kentucky. And Kibria has been there for a couple of years. And his background is in earth sciences, focusing on geochemistry and soil aqueous chemistry, urbanization and agriculture impacts on watersheds, associated climate change impacts, and better understanding the pore characterization and fluid flow behavior in porous media. So today he'll be talking about experimental investigation of tight rocks, which he'll be explaining a bit more in greater detail for environmental and energy applications. So Kibria, the floor is yours.
Okay, thank you, Maruti. So, so if it's okay, I'm sitting here and talking about, because otherwise I can't show some of the things closely. Okay, so here today I'm going to talk about these tight rocks and different type of experiments. And one important thing is, like, I'm right now working in a regional university, so we don't have enough resources. But within our like limited resources, we do a lot of research how we do that thing, and then collaborative things, and then what is the plan to use different type of DOE facilities with our current research. So that's what I'm going to talk about here. Okay, so at first I'm going to why these research are important. So one important thing is like right now, like unconventional petroleum development, and at the same time, like enhanced oil recovery. For that thing, we need to know about like what are the like tight rocks, what's their like porosity, permeability, like internal structures of these things, and then because depending on their like different type of scale chains, like whole flow system and their loss actually changes. So you can see when we have like mesoscale, that time we can use like Darcy flow easily, but when it is going to microscale, slip flow laws are present there. When it is going to more nanoscale, that time it's going to be Knudsen diffusion, other things are present. So that's why we need to experimentally understand the whole system, like what type of porosity, permeability are present there. What are the scales of all these pores? So that is the one of the reason behind doing these things. And obviously another reason is right now the carbon sequestration. So knowing our reservoir more better, at the same time, what type of seal we need, that is another important thing. And then, so I'm from an interdisciplinary department. So the like civil engineering also part of our department. So we also work with like different type of cementing material, characterization of those cement materials. So what are the, depending on like different type of mixture, how the concrete system is changed, like what are the porosity permeability system within the like cementing material, and how that can be like impacting the structure, like stability. So that type of things. And obviously another important thing is nuclear waste repositories. And actually, my PhD work started with nuclear waste repository works. So I started like analyzing samples for DOE nuclear waste repositories, different places, and then moved to like shale and oil gas related research. So that's the importance. So now we need to know also one important thing. What is the like basic characteristics of a tight rocks, right? So we know like most of our sedimentary rocks, or even some of the igneous rocks, when it is like volcanic igneous rocks, those have good porosity permeability. Most of the cases, those are in Darcy scale or millidarcy scale. But whenever we are going to like tighter rock, especially like shale or like tight sandstone, or even like more kind of tight sandstone, like this whole permeability is going to also almost like nanodarcy scale. And when it is going to narrow scale, so you can see like flow through in a 10-meter shale. In some cases, it take like fluid flow take like when it is like 10 to the power minus 7. Darcy scale, narrow scale, that time it's take almost like 300 years. Or when it is going to more and more, like almost close to like 10 to the power minus 10, that time 300 million years, that type of things. So that's why using these like permeabilities, we can also identify, okay, supercell zone for the permeability for like nuclear waste repositories, or like whenever we are any kind of contaminant we want to like preserve into any specific aquifer system, what type of seal we need above that thing. So that's why this is an important part of like what is the characteristics of the like tight rock. So here, most of the experiment, so we are going to understand what are the pore shape, their size, their pore throat distribution, how those things are changing. Because we saw that thing like based on those things, it's going to be changed what type of fluid flow behavior will show it, right? So that's why, like in general, we know these basic things who are from like this porous material characterization doing for a long time, most of you. So like we have pores, some of the connected pores. So we use different type of experiment to know about like their shape, size, at the same time, how they are connected. Okay, so what are their pattern inside? So in our porous media, so are those like, what is the tortuosity? What is the effective tortuosity of those things? And then how much pore volume are present there? So we use some simple technique, we use some like a complex technique. So here, so I'm going to show here some of the like experimental diagram based on these things, and then going to present like one by one different type of results, what we do. So one of the important thing when for our research, we collect the rocks. At the same time, we collect like well log, and based on the well log, we also get the data from the well log time well bore data set. Okay, what type of like porosity, permeability, other data set they got from when they did the drilling and different type of like data set. And based on that thing, we actually take our samples, and from that main core samples, we actually separate out the standard core size for our lab experiment for different type of experimental setup. So then we cut down that like samples, and then like decided what type of material we use for what. So some samples we use, some cases we use like cube shape size, and some cases we use like really craft samples for different type of experiment. And then so here I'm going to show like what are the different tests we do. So here, most of the cases, we do actually mercury intrusion porosimetry. So understand the like porosity, permeability based on like experiment, and there from the basic data set, we also use different type of equation and then get the permeability, tortuosity, those data set for the specific samples. And then also use FESEM to characterize like visually, okay, what type of shape, size, and other things are present there. We get the idea, and at the same time do the nitrogen absorption desorption process to understand, okay, what is the like monolayer or multi-layer present there, and then what are the inside shape of those pores for those things. And then another important thing for this type of work is understanding the weightability of the rock, because that is the another important thing with like whenever we do any kind of fluid flow related work, how our rock is going to imbibing any kind of fluid, or whenever we are doing any kind of oil and gas related production, how flow back water actually behave in our wells. So that is the purpose of doing like weightability and contact angle for that type of things. So we use both like different type of experimental method in lab for these things. So I'm going to show that thing. And then we also use a specific type of tracer and that tracer when goes through these like a specific core samples, and then we analyze those tracers using LAICPMS to understand, okay, how that like tracer actually moved through that all these pores, and getting the idea based on like concentration of these specific tracer. So how that whole like pore network is connected based on a specific chip. Okay, so so I'm just going to give some of the instrument a little bit of idea, and then going to show the their results. So in general, like marker intrusion porosimetry, it can be actually detect almost like till like 2.9 nanometer pores, the finest one, and we don't need actually below these one, because most of the cases like gas molecules, it's almost like methane molecules, it's in diameter is almost close to like 0.9 nanometer. And even like when it is like any pores, when those are like almost close to like 3 nanometer pores, Knudsen diffusion is more present there. So that is the another reason why we don't need a lot of cases, those things. So marker is like it's like fluid which we actually in high pressure we injected in our samples. So in this type of stem we use, and then we injected high pressure marker in there in the samples, and then in different pressure, how that like all these pores are invading by this marker. Based on that thing, we actually get the result, and we use all these different type of equations for getting others like result what we don't get from like machines directly. So marker intrusion porosity, we can directly get some of the result like in general result like porosity and then like bulk density or other things, and then different pores like different type of shape like length or other things, and we use that thing to identify like permeability and those things later. So here you can see from this basic graph, so how marker intruded. And then based on that inflection point, we also identify, okay, where the pressure is changing. So based on those things, we actually calculated, okay, how based on different pressure change, where it's actually injecting more and more finer pore system. And then based on those results, we get this type of data set where you can see, okay, so what is the total pattern of different type of pores and what is the percentage? So here I'm telling another thing. So in this presentation, I'm using 3 different type of shell samples, Woodford shell, Pearsall, and Hensel. So one shell, and some of the producing well, some of the non-producing well, and one is still on the development. So from the result, we also get the idea, okay, how these like pore throat diameter system is related with their production also. Okay, why this is important. So here in this like Woodford shell, especially where they drilled this one in the West Texas, permanent basin zone. So here you can see like most of the pores are actually a lot of pores, almost like a big number pores are within like this 2.8 to 5 nanometer scope. Okay, and so that's a like really tight rock for this zone. And then only few were actually above like 10, 50 nanometer zone. And we can see later like more detailed result of these things. So here you can see, and here based on their porosity, when we identify these pores, other results based on like SEM or other things, using those data set, we know, okay, when it is like less than like 5 nanometer, that means those are mainly like intercalated pores are present there. And when we do the like XRD for all these samples, we saw, okay, also they have like higher percentage of clay minerals. In some cases, some of have like clay minerals almost like 25 to 30%. And then you can see these pores are mainly based on like interpores, and then some are like organic matter. So I'm going to show that thing based on like SEM also. And then here is Haynesville shell, and then PSL shell. So I already told, so some shells are also, here is another shell, that one is producing right now gases, and from those from where we actually collected the samples for this Haynesville shell, you can see we don't have any like that much of like finer pores here. And then it has a lot of pores where their pore throat is almost like 10 to 50 micrometer, which is a good like size for flowing these gases. So that's why it's become like more better kind of production zone for this specific type of shell. And for PSL, so this is the current one in the development process. But the problem here, you can see like pore volume for these pores is really very less than other two. Like if we go back to like Haynesville, you can see like almost like some cases, some of the pores have like 30 millimeter cube per gram, or if you go to Woodford, it has also more, but PSL, it has less. So that's why still that one is in the development process. The PSL shell one is still that foundation is like on the like research phase. Most of the company are doing the researches on that shell. So here we can see a couple of more data for these things. So you can see here the pores in some shells, the porosity is high. And then another important thing for this pore throat network, we need to know, okay, what is the tortuosity? Because most of you maybe know about the tortuosity. So in general, like whenever we have like a straight 1 meter, so when we are taking the tortuosity things, so in the same 1 meter is becoming like almost 2100 meter. So that thing is becoming that whole network is more complex for this tortuosity thing. So, and then that's why it's important to check, okay, so, and you can see here, like in the Woodford shell, most of our samples, one thing is their median pore thread is really tight. Most of the cases close to like 3 to 5 nanometer, almost this is the median pore throat. And then they have like higher tortuosity. And when we go and check like for the Haynesville shell, you can see they have effective tortuosity. Some cases, those are really less. Okay. And they are also pore throat is bigger than 100 nanometer. So all these things actually connected with the production scenarios, how these things are related. So now another important thing is even we know like pore throat, which is the neck of the pores. So how the inside the pores are looks like. For that things we use like nitrogen absorption desorption isotopes. Okay. So when in pressure system we actually use nitrogen gas, and the pores actually absorb those and then releases those. So based on like how they're actually like getting all these gases and then releasing these things, based on that thing, so you can see the pattern. Okay, so when it's actually opening it, or they're like opening into when it is like in their desorption curve. So they have a specific type of pores. When it is closed, it has different type of pore pattern. And based on that thing, based on our data set, you can see all are actually 3 and 4. So that means these pores are based on like this type of system, especially pores like group pores, like this type of things, or like slit type of pores, or micropores are present there. And this like micropores are also all these samples are from Hainesville. And we saw like the Hainesville is the kind producing one shell where we from where we got the these core samples, and it actually easily give us the whole like idea also at the same time.
Can I ask a question on that?
Sure.
That's fine.
Yeah, sure.
And again, it's probably because I'm new to this field, like you're introducing.
Mhm.
But so the volume of absorption, the y-axis, it's centimeters per gram. That's the volume of the apparent volume of the pore, right? Is that right? The apparent volume of the pore in the context of how much nitrogen it's absorbing?
Yeah.
So as you increase the pressure, you're pushing the nitrogen into smaller and smaller and smaller pores.
Mhm.
Is that right?
No, for this one that didn't happen. So for the gas one, it's once actually get that.
It's just diffusion. Wait,
So that one is for the MICP,
okay, so
MIP.
just tell me, what is the volume of absorption? What is it physically? What is the volume of absorption? It's centimeters cubed per gram. So it's a volume per gram of soil. What is it?
So that's actually we are taking as a like in the pores, like how much gas is actually absorbed.
Okay, so I have a volume of nitrogen.
Yeah.
And right, so it's the apparent volume of the soil that the nitrogen is occupying.
Yeah.
And when you say relative pressure on the x-axis, what does that mean?
So so
You're changing pressure.
what is the general like normal pressure and how much pressure we are actually
So you are pushing the nitrogen.
pushing? Yeah.
So as you push it in, you're simply pushing it into different
Mhm.
pores,
Mhm.
and the increase is the volume. Okay.
Mhm.
Thank you.
Yeah. Thank you.
I have another quick
Mhm.
question. So why use only tight rocks? Like you don't use like limestone and things like this for these.
Oh no. We can use for any type of rock.
Okay.
Okay, so I'm showing this one for like for only for this one, but we can do this for sandstone, siltstone, clay, cement, for every kind of porous material. It's not like this is for only one type of things. We can do for any kind of rock, or even like
Soil.
we do these things for like soil samples. Okay. So now the another important thing is also like checking like images from the SEM, and then so it give us more better idea about like where the pores are and how the pores are present there based on these images. And you can see like whenever we actually do this type of SEM. So there, like, it gives us the whole rocks, both like uh, like mineral chemistry at the same time, like organic chemistry, both things. So here you can see most of the cases we have some of the samples we have like pyrite, premboiled, and then like intergranular pores are present within those pyrites event. And another thing is like we have like organic matters within these shells. So another thing why we do this SCM to understand like how much all these organic matter we have within a specific like area, getting that idea. At the same time, are those organic matter have like pores or not? Because that is also important thing. So we need to know, okay, there are like organic pores are present there. So that's why like you can see like these like organic matter, they got a lot of pores. And we saw also from our marker intrusion porosimetry in our organic matter in some shells, those have a lot of pores. And those pores, in some cases, those are really tiny. And you can also see here, because our imaging power wasn't that enough for this SCM. So here you can see all these pores are really tiny, even very smaller. But some pores, like for these porous organic material, you can see these pores are really larger. So that the thing we actually use the SCM to understand all these different rocks, like how the pore systems are related.
That scale bar is nanometer, 200 nanometer.
Yeah.
Even the not organic matter pores are really tiny.
Yeah, that's the problem with the shell. So some organic matters, even some like interparticle pores or intra particle pores, both are most of the cases very small. So that's why we do like hydrofracking. That is the another thing. That's the reason behind this.
How do you define mineral pores as opposed to intercrystallized pores? What's the difference?
Sorry?
How do you define mineral pores? What is that?
So these are the clay minerals. So within the clay minerals and then other minerals, other pores are present there or not. So also for these things, so when I show the marker intrusion porosity, so same thing we also did for all these different type of minerals, like for quartz, for like different type of clay minerals separately, for calcite, and then what these different minerals have different porosity, we also separate out that thing. And that one we also use in some cases. So I have those data also, so later I can show that thing.
But so minerals are crystalline materials, and you have these intercrystallite pores. You're trying to understand how they are different.
Yeah, so when it is totally intercrystallized, so that time they don't have. But when they're like dissolved, some kind of things are present there, maybe some calcite minerals, it got like dissolution somehow, or some other type of things, or like clay minerals. So that cases, those are those clay plated pores. So those things.
So it seems like it depends on the crystallite size or the crystallized structure,
Yeah.
grain structure,
Yeah.
because from the picture, it seems like mineral pores are within a bulk piece of mineral, and the intercrystallite ones are when you have these crystalline grains and the pores between them.
Yeah, so like here we don't have anything like totally crystallized. So somehow like within the crystallization phase, somehow we got this tiny like intercrystal system, like a small pore.
Yeah, sorry, thanks.
Your question is perfectly fine. Now the another thing is we do like spontaneous imbibition. So in our lab, we also used like this type of simple setup to get the like how like water is going to imbibe from a like shell core, and from that thing getting the idea, okay, how the like population threshold system is working there, and how we can use those things for other calculation. So for those things, we use this use this experiment. So here, the thing is, you can see like we use this type of chamber. So within this chamber, we try to avoid any kind of like excess kind of evaporation things. So we try to like avoid that thing, and then we use a specific type of hook, and based on that thing, we placed a sample, and that sample actually one is actually exposed with the water, and other sides normally in general we actually epoxy it, so that's why it will not get the excess water from the sides. And then how much water that is actually collecting using a like wedging scale, we actually get that data, and then in a like say this type of computer setup, we actually get that whole number every minute. Okay, and
They're from cores.
yeah, course, the course there. So that's why we cut the course in like some are like 1 centimeter dia or some are 2 centimeter dia core. We actually plug that type of core separately, collect, and then use those things here. And then like using that thing, so what we get like a how these all these water is imbibing. So what is the purpose of these things? So this whole imbibition system slowly give us, okay, what are the based on different type of rocks, like characteristics, and their all this pore connectivity, how much water slowly they can imbibe. So, and then at the same time, when we actually analyze those things based on those data set, we can get, okay, their imbibition slope, and this slope's actually directly related with the like population thresholds things. So like if the number is above 5, then it's a high imbibition system are present there. And then for these things, another important thing we did, like we use several different type of fluid. So some are water, some are like API brine. Some cases we use like other fluid like oil, and then see, okay, for different type of fluid system, how all these rocks shows their imbibition like process. So we can later use that data to correlate with like weightability. So for the weightability, so we use like this tensiometer sometimes to get the direct contact angle with the from the sample surface after when we drop a droplet of like a specific fluid from that one. And sometimes we also like qualitatively we do like how this like non-weighting or weighting based on like images, processing the image, like how fast it's actually weighting the surface of that sample. So based on that thing, it's also like give all this result, okay, for a specific type of fluid, how that specific rocks are actually hydrophobic or hydrophilic, what type of behavior that rock has, and are there any relationship with all these like different type of pores with their hydrophobic behavior and or with the like minerals what are present there. And then based on that thing, we can also like get the idea, okay, how this production thing is also related. So here you can see like for API bind, so all these like, this is the Woodford shell, they have really higher contact angle. But when you can see like this Haynesville shell, they have like moderate contact angle. So there's an internal like waiting with this like fluid. So that means even like imbibition process easily can like work there when we are like doing any kind of like flowback fluids are like we're trying to flow back, or whenever we are trying to like do the hydrofracking, that type of things we can do easily. So that type of results we also combine with these things. So another thing in our lab, we built this method to do and understand easily in our limited resources to understand like pores and then pore connectivity using the vacuum saturation. So we build these things. So I'm going to show our apparatus later. So in general, we try to vacuum the whole chamber, and then we actually flush it with the carbon dioxide, and then we injected fluid. And then with that fluid, we actually use a specific type of known diameter tracer. So that's where we can know, okay, how and what type of tracer are actually passing through these samples pores. Okay. So that's why we use a specific type of iodine and uranium based tracer. And then using LAIC pigments, we actually get the whole concentration of the sample surface. So here, this is the setup. We actually using our machine shop, we built this one for the vacuum saturation. So here we use still like 50 psi to vacuum the samples and then pressurize, and then using this specific type of size tracer so that we know, okay, where we are getting this tracer concentration, how much and where it's flowing through our like this experiment. So for these things, what we do, so after this vacuum saturation, we use high pressure impregnation using like mercury emission porosimetry. And then when all these traces are actually going inside of the sample, then we freeze dryer that whole like all this tracer, and then we actually use LAIC PMS to using the laser ablation card, and then getting the solid samples, and then analyze their concentration in our samples in different depth even. So we can actually using like LAIC PMS, we can also based on like laser ablation, we can change how depth we can go and how like it's changing. So sometimes we also do slicing the samples and getting the data from that thing for the concentration. At the same time, using this vacuum saturation, MICP, all these samples, other things, what we do, like we can actually compare their like porosity permeability based on all these different type of tested. So how these things are changing and what are the differences are happening. So when we get the samples from the from our well field, so that time based on the core log, we got one set of data, and then from different type of experiment, we got all this type of data, then we can actually compare those data set together. So so finally, so when we actually analyze all these tracer, or using ICP-EMS, then we actually use these images to get the idea, okay, where and how these things are passing through in our samples. So here you can see this is the surface. These are the surfaces, and these are the top surface, bottom surface, and these are the internal after cutting it and then getting the concentration based on ICP-EMS. You can see only the edges, those pores are connected. So whenever it's going to more inside, only few places it's getting these specific iodine tracer here. And the same thing for the rhenium. You can see it's actually in these cases we get less of this one because these things is also related with our what is the, so this is more a smaller, so that's why we are getting these things here more easily, most of the accessible pores. But for this one, it's more, for the rhenium, it has more a higher size. So that's why only in the surfaces we are getting. So we also use this type of tracer things for for any other rock types to getting how can also any kind of contaminant can pass using this type of ICP-EMS. We can also analyze that thing. So yeah, so overall, the thing is, this type of experimental understanding we use to identify tight rocks, characteristics, and then easily we can get porosity bound to all this behavior, and then with all this low cost equipment in a regional university, this type of experiment we can do. And obviously, all these type of experiment we can use for all these different type of field, not only oil and gas, at the same time carbon sequestration or nuclear waste repositories, groundwater contamination, all these things. So far what I'm lacking, so I need to do NMR, XCD, and then a small angle neutron scattering. So all these things will be getting more better fit with all these results, and that will be more important. So another thing is important thing. So all these pore-related resources developed more with the shale revolution, which is actually 15, 16 years right now. But so far, important thing is we need to know how one specific formations, all these porosity, permeability, tortuosity, production, we can combine together and do some kind of machine learning work to actually know the best result, how we can do for this type of not only research, but also apply those things into the field level. Okay. Yeah, thank you. To answer any question and any suggestion if you have to change anything or develop anything. Yeah.
Just one of your earlier slides when you had this plot of intrapore and interpores.
Oh yeah.
Is it equivalent to connected and unconnected pores, or means something different?
Where?
It was in the beginning of the talk.
Oh, beginning. Okay.
It was, this column diagram with interpore.
Oh yeah, sure. Got it. This one.
Yeah. So just how do you define intra and inter?
So in general, we are defining this one based on our SEM images. So in general, in SEM images, we see in between the grains, where the pores are, those are in general like above like 50 nanometer. But most of the cases like intrapores, we are seeing those things like within this range. So based on that thing, we actually
So
classify.
it's image-based, you basically.
Image based, yeah. It's not nothing we can tell based on our experiment directly.
Just checking.
Yeah. Based on our experiment, obviously it's not going to tell us, okay, how that pores are present there. Yeah, so it's just like correlation with the like how we are getting images from our SCM data set.
Okay.
Any questions online?
Is there questions on the chat?
Let me check.
So what is your plan for XCT, for example?
So,
You
so
want to integrate?
yeah, the same samples, what we are like experimenting these things. So I need to know about that specific like 1 centimeter core plaque, okay, from the XCD where the pores are present, and then what type of things are changing into the XCD, that 3D images. So we can then more better use like what are the relationship with our like when we are doing LAICPM stress surveys or other things, are those we are getting the exact letter or result or not. From the XCD, we can actually more compare those things.
Yeah, it will give you the 3D context and
Yeah,
but
yeah.
you know the resolution is different, so it's limited.
But
It still can be
it still can be, we can
helpful.
correlate those things.
So can we get these kind of pore, because these are nanometer pores?
Yeah, we can't. So that's I'm saying that kind of complementary almost, but if you can look at the larger pores, that those you can see based on sample size in both methods, but you don't see nanopores or even submicron.
Yeah, so that's why for the nanopores, most of the cases we do the small angle neutron scattering. So yeah, but that is not.
Yeah.
I don't think you guys have that one here. So these.
No, but yeah, there are some people in Oak Ridge who are.
Yeah, yeah. Okay, they have that one, and then NIST they have that thing, National Institute of Standard and.
Question.
So, it's kind of an interesting problem of not being able to see with some tools like XCT, some of the pores that might be important, right? That's kind of the challenge.
Mhm.
Like each of those two methods probably has their own benefits.
Yeah.
And as I saw the one slide, it looks like a fair amount of the porosity is down in the smaller pore sizes in some of these systems. So there's a parallel problem in the lungs when we try to map the pores in the lungs to better understand fluid flow, airflow, toxicant flow.
Yeah.
Some of the pores are really small and it's hard to actually map that structure. So here's my question. What I'm wondering is whether or not if you could more conveniently, more commonly measure things by XCT and get the pore structure up to, it's like micron scale.
Yeah.
Yeah. Is there information there, enough information there that allows you to actually successfully predict the pore structure for pores that are smaller, right? I mean, because you could imagine, I could imagine that just given the compression of the system, the packing and all that, that if you know what the pore structure is up to a micron, there it is, you have all this information, that actually is pretty informative about what the pore structure is going to look like in these smaller pore sizes. Maybe I'm completely wrong, but has anybody tried to do that? Where you have information on both pore sizes? Can one be used to predict the other? Ooh.
Yeah.
Is it first of all?
I will comment, but I want you to go.
What is the exact question?
Oh, sorry. So the exact question is, if I had a data set with the entire structure of the pores from big to small, and I had 1,000 of those, could I build a model that used only the structure of the pores up to a micron to predict properly, accurately, the pore structure of everything below a micron, that you have to use some of these other techniques to measure?
Only one test, it's difficult. So we have to use a couple of different, because one test will not give the exact one specific. So all the tests have some limitation. That is the problem. So that's why, other than using all different type of tests, it's kind of difficult to do that thing.
Lots of good things are difficult. Tomasz?
Well, my thought was that obviously these are different scales, and you might be able to figure out or model a structure, but the modeling part really depends on the question, right? Because at the nanoscale there'll be different forces like capillary forces and molecular interactions play an important role. While in a millimeter size, micron size pore, it's maybe the water will just flow more readily. So it's different interactions, different drivers, I guess. But
So that would be modeling the behaviors of liquids and things in those. My question, which apparently was poorly framed, was maybe a little bit different than that.
Suggestion.
yeah, but
The thing is that like it's the pore size and also the pore connectivity. So if you look into his last one, the vacuum part, so you can see the outer regions are connected. The inside it is not connected. So even small pores, even if we characterize a lot of small pores, if they're not connected, they might not have that much of value. That's the reason why they're breaking the rock to make them connected. So even though if we have information until the micron scale, if you want to predict what's gonna happen below that, even if you use any sort of machine learning or any other thing to even super resolve what's going on below, the goal is like if they're not connected, we might not have, like, that might not be that useful when you're doing a flow experiment or when you're trying to not break a rock. So in the rock context, you need to break it to make it accessible. In the soil, you don't need to break it. It is conceptually accessible. Correct me if I'm wrong, Thomas. Like,
yeah. I also wanted to add to my comment that it may not be useful depending on your research question to include those nanopores, because they are so insignificant in volume and they don't transit, but they are very important for something like
There's like
I think.
organic
Okay.
matter in the
Yeah.
nanopores.
that's why like
yeah
there's a lot of organic matter.
Yeah, another problem is also like,
we don't know them.
what is the pore size? Depending on that thing, like whole governing equation system is also the chance. So that is another problem. So like when it is going to micropores or macropores, it's going to be like, we can use Darcy's law. And then when it is more smaller, then it's going to be like slip flow system. And then when it's going more smaller, then so that is another problem in a one single system. If there are like all these different type of pores, and if I want to use all these equations at the same time for one single, I don't know how that thing will work.
So in
I have-
that graph you showed of different pore sizes,
Yeah.
do you know if there is a relationship of a particular material of like maybe if you have the limestone versus like a shale rock, then is there a difference in?
No, there are obviously differences. So all these different rocks, it will be changed and separated. So that's why like when it is shale different characters. So when we take limestone, like they have all this system will be changed.
So maybe then if some of these type of rocks have only pores up to a certain diameter.
Yeah. So these things only happening only in the shale or tight sandstone or siltstone. Yeah. For nothing else. For if we'll go to like other things, it will be chance.
Question, like very naive question. When it comes to soil, the pore connectivity is constantly changing.
On the soil?
Yeah.
Yeah, because the compaction is going to change. Even after a big rainfall in the soil, the compaction will be changed. So the connectivity and pore system will be changed.
But yeah, back to what Justin was also talking about. It's really interesting to see those nanopores. What would be interesting to know more about them. And so one thing is that we people refer to this as core scale research and also aggregate scale or tiny. And so we are interested in, for example, let's say look at a small tiny soil aggregate using TEM.
Mhm.
You can see the pores and the organics and minerals and see if that knowledge can be included in models,
Mhm.
concepts or something that will then connect to the core scale. But yeah, it's
He's planning to write a LSR this year, so he's encouraged to the full proposal. So
yeah, cool.
he's developing techniques to like from the modeling perspective. So one of the things he's interested in, they have
Yeah.
all the data sets related to XCT and SEM-EDX. So how we plan to link that up.
Yes, since there is no one technique that can see both scales, models are
Yeah.
going to be very critical to bridge. Okay.